Hey guys, today we're going to be discussing Chapter 4, Important Lab Media. Now, culturing microorganisms in the lab requires an appropriate culture media. Now, keep in mind, remember that the majority of prokaryotes that exist on planet Earth have never been grown successfully in a lab. However, we do have some culture media that we know work for certain organisms. We have general purpose media, selective media, differential media, and some media that are both selective and differential. Now, a general purpose medium is used to grow most lab organisms. We use it to determine colony morphologies, so something like tryptic soy agar is a good general purpose medium. Lots of different commonly used lab organisms grow on it. Selective media inhibits the growth of some bacteria while selecting for the growth of others. So an example here is a medium that's selective based on its pH. So in the neutral media on the left, we see lots of different types of bacterial colonies. But in an acidic medium, we're selecting for the growth of fungi instead. Differential media differentiates between two different organisms growing on the same plate. For example, we use blood agar plates, so a tryptic soy agar with 5% sheep's blood in it. It's used to differentiate between different hemolytic abilities. Alpha hemolytic organisms have an incomplete lysis of the red blood cells in the sheep's blood, and that makes them appear to have a green zone around their colonies. Beta hemolytic organisms are able to completely break down the red blood cells, and they have a clear zone around their colonies. Gamma hemolytic organisms have no lysis of the red blood cells, and so no change to the media. So if you look up here, you see the green zone around the alpha hemolytic, the clear zone around the beta hemolytic, and the gamma hemolytic down here where there's no change in the red color around the colonies. All right, and selective and differential media. This would be something like mannitol salt agar. It's selective for only salt-tolerant organisms can grow on it because it has a high salt concentration. About 7.5% inhibits the growth of most bacteria. And it's also differential from mannitol fermenters. Mannitol is a sugar, but not all organisms can utilize this sugar. It also contains a pH indicator. So this turns the media yellow when the mannitol is fermented due to the acidic byproduct that's produced. So organisms that can change the color of the media to yellow here, we know are able to use that mannitol sugar, and they're salt tolerant. So if you look up here in the top left, here's a clean mannitol salt plate that has no bacteria on it yet. And then down here in the bottom right, we can see bacterial growth on both sides of the plate, but on the right side, these bacteria are able to utilize that mannitol sugar, whereas those on the red side cannot. Another example of selective media here is Eosin methylene blue. These EMB plates are selective for gram-negative coliforms. Eosin and methylene blue inhibit gram-positives, while also serving as a pH indicator in a sense. The rate of lactose fermentation makes this media differential for two major coliforms, E. coli, which shows up as a metallic green, and all of the other coliforms, which are the purple colors. So EMB is both selective and differential. All right, and next we're going to talk about biochemical testing. So every bacterial species produces different enzymes, which allow them to do different metabolic processes and create different metabolic byproducts. This means that not all bacteria are created equally. For example, not all bacteria can metabolize lactose. So, for example, here we have the results of a Simmons citrate test. So the blue tube on the left shows a positive result where citrate is metabolized and it changes the pH so the tube looks blue. The bacteria in the tube on the right are not able to break down citrate, and so the tube remains green. Another test we do is the Gram stain, or another type of stain. The clinical significance of this is it allows us to make more appropriate antibiotic treatment selections. And it's the first step in aiding in identification. We can rule out an entire group of bacteria and allows you to choose the correct biochemical test to follow up with for identification. You guys will be doing more of this later in the semester when you work on your unknown bacteria labs. All right, so the Gram stain. First thing you'll do is your slide will be flooded with crystal violet stain for 1 minute and then rinsed with water. After this stage, all cells are stained purple. The second step, the site is flooded with iodine for 1 minute and then rinsed with water. Iodine acts as a mordant. It helps to lock in that purple crystal violet stain, and all cells remain purple. Now, the third step is the slide is flooded with a solution of ethanol and acetone for 10 to 30 seconds and then rinsed with water. And the result is that the smear is decolorized. Gram-positive cells remain purple, but the gram-negatives are now colorless. We've washed the color out of the gram-negative cells. The fourth step is to use a secondary stain, and the slide is flooded with safranin for 1 minute and then rinsed with water and blotted dry. The gram-positive cells remain purple, but now the gram-negative cells are pink. It's important when you do a gram stain that you're using young cells that are less than 24 hours old. The peptidoglycan begins breaking down, leaving you with a mixture of pink and purple cells, even from a pure culture of gram-positive bacteria. Another important thing to keep in mind is don't over-decolorize. This is the most important step. If you accidentally over-decolorize, your gram-positives can appear as gram-negatives. Usually you'd end up with a mixture of pink and purple cells. All your gram-negatives would still, of course, look pink. All right, so that's it for media and lab techniques. You guys will have a chance to practice and apply these ideas in the lab.